The first type of vessel we're going to examine are large elastic arteries, such as the aorta. These are quite wide in diameter, and they contain both a lot of elastic lamina in their wall, as well as maybe 50 layers even of smooth muscle cells. So they're quite large, quite thick-walled structures. But let's start at the inside and examine the features that are common to this type of vessel. First off, they have a relatively thick tunica intima, which means that in addition to the endothelial cell lining, they have quite a lot of connective tissue underlying that. And they have a very thick and well visualized internal elastic lamina. They also have a very thick tunica media with many layers of elastic lamina, and that can be seen on the micrograph here on the right, which is an elastic stain, and you can see all the black elastic lamina. Looks like all the little wavy lines. The tunica adventitia in this vessel contains vasa vasorum as well as nervi vascularis, and is usually then outside of the collagenous connective tissue has a layer of fatty tissue, more or less for some insulation kind of shock absorption. So here's a H&E looking at the aorta, and here the elastic lamina appear as the pink wavy lines. So this is not an elastic stain, but what's really nice here is I think you can see how relatively thick the tunica intima is here. So you can see the endothelial cells pointed out as well as the underlying connective tissue, and then you begin to have the elastic lamina interspersed with the smooth muscle cells, which really aren't visualized all that well in this image. Then you get to the external elastic lamina at the outer edge of the tunica media, and you have just a little portion of that tunica adventitia, which is primarily comprised of collagenous connective tissue. Muscular arteries are somewhat smaller than the large elastic arteries, and they feature a very prominent internal elastic lamina, which can be seen on the right here as that very dark, variegated looking line. They also have discontinuous elastic lamina within the tunica media. What else can you see? Well, you can see from that picture that the tunica intima is very difficult to visualize. So it's much thinner in a muscular artery than it was in the large elastic artery. The number of smooth muscle cells in the tunica media can be anywhere from 3 to maybe about 40 or so. There's no need to count it. You really just need to look for that internal elastic lamina to define this as a muscular artery. Sometimes you can see a very clear external elastic lamina bordering the outside of the tunica media, as in this diagram. And outside that would be the connective tissue of the tunica adventitia. The medium or larger sized muscular arteries may have vasa vasorum and nervi vascularis of various sizes. As you get into smaller and smaller muscular arteries, those will be less evident. So here's a higher magnification showing that wall, and it really points out the thinness of the tunica intima with the very prominent internal elastic lamina, and then the thick tunica media. Those smooth muscle cells, remember we talked about those in our last unit, and I said when they're contracted they often look corkscrew. So that's a very common appearance of the smooth muscle cells in the tunica media, which are circularly oriented around the lumen. As you go down further and further in size, certain features become more or less prominent. The next sized vessel is called an arteriole, And arterioles we're going to define as having no visible internal elastic lamina at the light microscopic level. Now, if you look at these with an electron microscope, you can sometimes distinguish an internal elastic lamina, but not at the light microscopic level typically. So we're just going to say that they lack it. They also have, in the tunica media, only 1 or 2 smooth muscle layers, and then they have a very small tunica adventitia that really just blends into the surrounding connective tissue. Here are some good examples of arterioles. Again, they feature a very thin tunica intima, no apparent internal elastic lamina, only 1 to 2 smooth muscle layers, and a thin tunica adventitia. Capillaries are the smallest diameter vessel. They comprise the microcirculation. The lumen of a capillary is often not much bigger than the red blood cells in it, somewhere around 8 to 12 micrometers in diameter. The wall is a simple endothelial tube, basically just a tunica intima. Some capillaries have a tunica media comprised of a pericyte, which is a specialized cell that I'll discuss in a few minutes. There's no smooth muscle, so the tunica media does not exist in the same form as it did in the other vessels. The tunica adventitia essentially is just the surrounding connective tissue, so there's no real layering in these microvessels. Here are some examples of capillaries directly underneath an epithelial layer in the connective tissue underlying it. And you can see that each one has a single endothelial cell nucleus in the plane of section. Oftentimes that's the case because they're so small, one cell wraps the entire lumen at that particular point in the section. So let's talk about pericytes. What is a pericyte? It is actually a connective tissue cell similar to mesenchymal cells that we studied when we looked at general connective tissue. It surrounds a capillary endothelial cell within the basal lamina, and it provides a little bit of structural support. It also has some contractile ability, so it acts as in the same manner as a smooth muscle cell, but it's not a muscle cell per se. It participates in physical and chemical signaling with the endothelium, and in some cases may give rise to endothelial cells or smooth muscle cells during wound healing, and that's the process of angiogenesis. So capillaries can be broken into 3 big types. The first is called continuous capillaries. These capillaries have tight junctions between all of the endothelial cells and a continuous, rigorous basal lamina. The second subtype is called fenestrated. The fenestrated ones actually have holes in the walls. They do, however, have a continuous basal lamina underlying the endothelial cells. So fenestrated can have diaphragms, in other words, molecular coverings over the diaphragms, or they can be without them. The third type is called sinusoidal or discontinuous capillaries. These capillaries are very large diameter lumens, and they're like dilated capillaries basically. They have 2 features that differ from the other 2 types. First of all, the cells are not generally completely adhered to one another with tight junctions. In other words, there's literal spaces between the cells that line the wall. Second of all, they often have a discontinuous basal lamina. These are found in places where you need a great deal of exchange between the blood circulation and the underlying tissue, such as in the liver. You see this kind of sinusoidal capillary. Also in bone marrow. Here's a 3D view that hopefully will make things more clear. You can see how the cells in the continuous capillary in the endothelial cells are all tightly adhered to one another right up against with no spaces. And then you can see the fenestrations in the fenestrated capillary, and there may or may not be molecular sieves acting as diaphragms over those. And in the discontinuous capillary or sinusoidal one, you can see that there's actual large spaces between the cells.